Hello, my name is JonathanDavid, and this is my presentation on profiling algorithm for colorblind simulation and visual aids. The purpose of this project is to help with the simulation of anomalous trichromacy. The problem with most online simulators today is that they actually only simulate dichromacy, which is the complete lack of one of three cones. Those that do allow for simulating anomalous trichromacy only give one option for each type of deficiency. The problem with this is that anomalous trichromacy exists among a range, not one single deficiency. So I'm trying to make a tool that will simulate the full range of deficiencies for the user. So to understand color blindness, you must begin with the basics. So there are three photoreceptors in the human eye. There's the L cone, which is responsible for perceiving red light. There's the M cone, which is responsible for perceiving green light. And there's the S cone, which is responsible for perceiving blue light. The range of our color vision is a result of these three cones working together. For example, yellow light would be a simulation of the short cone and the medium cone to perceive yellow. Color deficiency occurs when one or more of the cones have defects, such that you cannot perceive the color as well as someone who has all cones functioning normally would. Anomalous trichromacy occurs when one or more of the cones has a defect and the sensitivity to that light of the spectrum is shifted. There are three basic kinds of this. There's protanomaly, which is a defect with the L cone, deuteranomaly, which is a defect with the M cone, and tritanomaly, which is a defect with the S cone. Protanopia is a condition where the L cone is completely missing, deuteranopia is a condition where the M cone is completely missing, and tritanopia is a condition where the S cone is completely missing. When one of the cones is totally missing, it is called dichromacy, but only one has a defect and is present but does not work as well as it should. That is called anomalous trichromacy. So the figures on the bottom of the page represent kind of what happens when you have anomalous trichromacy. The figure on the left shows protanomaly, so that is your L cone has a defect. You can see that the band is shifted slightly to the left, and it's more overlapped with the green and the blue cones. The middle figure shows deuteranomaly, which is a defect with the M cone, and you can see that the M spectrum is shifted slightly to the right, such that it is now less overlap with the S cone area, but more overlap with the L cone area. And the right figure shows tritanomaly, which is a defect with the S cone, and you can see that it's shifted to the right, with more overlap on the L and M cones. This overlap is actually responsible for the decreased perception of color vision, because the way our eyes actually work is we'll take a difference between the L and the M cone, and that will show us a different kind of color. Well, if the band passes are more over each other, the difference between them is less, and therefore you're having more trouble perceiving that color. This is an image of the human fovea. On the left there's normal human color vision, and on the right there's protanopic vision. You can see that on the right the red cones are completely missing from that image, and that this person would not be able to perceive a red light at all. The project goals were to accurately recreate a color deficient image, to allow a user to save a profile of a certain type of deficiency and apply that profile to a different image to recreate their color deficiency for a different image, and to shift the colors in an image so that a color deficient individual can see the differences in colors that they would otherwise miss. So basically you would take a color that they can't see, shift it to one that they can, and just kind of accentuate that color so that it pops out more in the image for a colorblind user. Simulating color blindness begins by first transforming the RGB spectrum into the LMS domain. So the LMS domain is actually how our eyes work. RGB is a combination of activations of our different cones, so it's more intuitive to work in the LMS domain, and that way calculations become easier to perform in the LMS domain, since that's actually related to how we view images. The image on the right actually shows the mapping of the LMS domain to the RGB domain, and the Q lines actually show something called lines of confusion, which would kind of map what colors a color deficient individual would have trouble seeing. So recent studies have actually tackled anomalous trichromacy. So previously they were only tackling dichromacy, which was the problem. So the introduction of the two-stage human vision model has helped tremendously with that. Basically, it has used research on human vision to determine what we actually see, because again, if you've been colorblind all your life, you're going to perceive colors in a different way, because your brain will kind of adapt to your deficiency. So the second major step would be transformation from the LMS domain to the opponent stage model shown on the bottom of the image. And the purpose of this is to more accurately represent what a human would see if they have color deficiencies. So the final step is to derive a transform matrix for colorblind vision simulation. Matrix calculations are fast and easy, and once calculated, the algorithms can be run in the RGB domain. So basically, you perform a lot of complicated math to obtain a transformation matrix, and then that can be applied directly to an RGB image with just one matrix multiplication, which is much faster than recomputing it over and over again. So a quick rundown of the math from going from RGB to LMS. You first take the DAC values and transform them to RGB. So basically, the computer doesn't work in just displaying red. It has to have a DAC value sent to the monitor. So you take this DAC value, divide it by 255, and raise it to the power of 2.2 to get a certain floating point number. So the second image actually shows the result of mapping RGB to XYZ, and the third image shows XYZ to LMS, and then the final shows the final matrix calculation what we use to obtain the RGB to LMS calculations. The numbers shown here are actually obtained from previous studies and from certain documents that tell companies how to map colors on a computer monitor. So it's a certain protocol that they have used to obtain this matrix. So the second thing you do is go from LMS to two-stage. So you have to shift the spectral sensitivity, obtain two-stage parameters for RGB colors, and obtain a color-deficient transformation matrix. So here in the top image, you can actually see the LMS being transformed into the two-stage model. The second image shows what happens when you have a certain color deficiency. So delta sigma L is basically saying you are shifting what someone sees from the spectrum slightly over to the right. The third image shows how you obtain the three different factors of the two-stage model for each red, green, and blue. And the final shows a matrix called gamma that would just encapsulate all of these and be used as a transformation matrix for either a color normal individual or color blind individual. So the final stage is profiling and Daltonization. So basically the end result of all the work is just one matrix. Gamma is the color deficient matrix used to obtain color transformations. So this is the profile that we would use. We basically reuse this matrix over and over again for different images. And M represents the Daltonization matrix used to shift colors in a spectrum that a color deficient individual can perceive. So basically this is taking a color that they cannot perceive and shifting it to one they can, and it basically is summed with the original image, kind of making certain colors more apparent. So the results of the study were a success. It accurately creates a color deficient image among a wide range of anomalies. It allows users to save a profile of a certain type of deficiency, and it shifts the colors in an image so that a color deficient individual can see the differences in colors they would otherwise miss. So the three results are on the bottom. They are deuteranomaly, protanomaly, and tritonomaly. The top rows for each image shows the colorblind simulation of a color wheel, and the bottom rows are the Daltonization of that image that would help a colorblind individual perceive the color wheel in a way that we would. It wouldn't necessarily give them the ability to see the color, but it would help them differentiate the colors from each other in a spectrum that they are familiar with. So as you can see for deuteranomaly, the reds and the greens kind of slowly become more green and harder to perceive differently from each other. And the Daltonization of the image kind of makes them very radically different, or changes the shades to something very strong that they would be able to perceive. For tritonomaly, while the reds and the yellows, well, the reds mostly stay the same, and the yellows kind of fade, and everything becomes kind of a more purple grayscale. And for deuteranomaly, the greens and the reds are shifted. Protanomaly and deuteranomaly are actually kind of really closely related to each other, and that becomes hard to perceive red and green apart from each other. So this is a peanut butter example. Basically, this is the center image shows what a protanomalous user would see if they have a 10 nanometer shift in their cone vision. The original image on the far left shows what a color individual would see. The CVD image is the simulated result of what they would see, and the Daltonization image would help them perceive the differences in colors in the image. You can see that it's kind of more bright, but I myself has protanomaly, and for me it helps me see the differences between, the color of the peanut butter and the color of the bread, because to me peanut butter itself looks green, and that is not correct. So on the Daltonization image, I can actually see that everything is about the same color, and that is how a color individual or color proficient individual would see a peanut butter smeared over bread. This is results for a deuteranomaly of about 4 nanometers on apples. In the original image, you can see that there is a mix of red and green apples, and in the color vision deficient image, you can see that the red actually shifts a little bit more towards green, but the green apples are left completely unchanged. And the algorithm basically does not try to shift colors that are not affected. And in the Daltonization, you can see that it takes the red apples and shifts the colors so that it would help a anomalous person perceive that those are actually red apples, not green apples, and separate them from one another. So, since part of the results were to show the range of color deficiency, I have an image of flowers in shifting from completely normal to fully trichromatic, and you can see at each step the color transformation is actually more and more severe. So the first image is completely color normal, the second image shows a 2 nanometer shift, a third shows a fourth, the fourth image shows a 6 nanometer, the fifth image shows a 8 nanometer shift, and the final is complete basically trichromacy, where you cannot see blue light very well at all. So here are the references. I use the first paper and the second paper mostly focus on dichromatic vision, and the third and fourth paper have extensive research into anomalous trichromacy, and they contributed a lot to this research. Alright, so I'm going to now show you a working simulation of how you would use the GUIs to come up with a color profile for a colorblind individual. It begins by opening the test GUI, and you can see that you can pick a deficiency, either protonomaly, deuteranomaly, and trianomaly. I have a traffic light example preloaded for this. So in protonomaly, you can shift it to, let's say, about half severity, and you can see that the color here went from red to a little bit more green, and that I'm actually colorblind, and I can't perceive a difference between these two bottom images at all. For me, over here, this is where I can't perceive a difference between the two colors of the two red signals, and a completely protonopic individual would see this image. So if you go over here and change the deficiency, you can actually switch between protonomaly, deuteranomaly, or trichromacy, and you can see that trichromacy again kind of perceives red and has a shift in every other color. So if you wanted to save this profile, you would go to Save Profile As, and let's say this is a trichromacy. Hit OK, and it will save a trichromacy.mat file over here that can be reused to recreate the same effect on different images for different people. You can also choose a different image, so let's say if you don't want to use a traffic light, you want to use a color wheel instead. You would just type in the name of the image you would want, put it in, and then a different image would pop up. This allows you to select a different image that would more accurately kind of capture the point at which you can no longer perceive the color change, so that you can know about where on the spectrum you lie. Alright, so with this GUI, this creates the profile, and I created a different GUI that would show both the original image, the color deficient image, and the Daltonized image. So we're going to open that next. It's called CVD Display. Alright, and right now it's showing a peanut butter smeared on bread image that I showed previously, but this is a color normal individual, so all three images are not being shifted at all. Again, this profile is assuming that you have a normal vision. It's actually resolved with normal v.mat over here. So if we wanted the profile that we just created, we simply go over here and type in the name of the profile we just saved. And you can see that the colors are now shifted between normal vision, the color deficient image, and the Daltonized image. Again, you can choose a new image, so let's say we wanted to see apples, for example. And now we can perceive how a blue weak person would perceive apples. And you see over here that the Daltonization actually shifts it towards green, and this would help them see the difference in colors, whereas to us, it looks kind of very odd. Let's try making a different profile again, and one that is not trichromatic. Let's say I want to simulate a protonomaly of about medium, light severity. So we're going to save this profile as prot. Alright, it's been saved. And let's go back to CVD Display. And let's use this on the color wheel image and say that we want it to be prod. Oh, okay, so for me, I actually didn't perceive the change in these two. I was kind of looking at the center, waiting for it to change. And the Daltonization kind of came up, and that's what caught my eye. So you can see in the left, this is the color normal image. On the middle, this is the protonopic image. And on the right, this is a Daltonization. This is helping me see that there's a difference in the range of colors over here. Whereas here, it just kind of meshes, kind of molds together, and I can't perceive a difference at all, whereas here the lines are very, very clear. Alright, well, that is my presentation. That's my project. I think it was a success. I can clearly see that the original image has been shifted. It displays the range of color blindness among individuals kind of smoothly, and that the Daltonization image helps a colorblind individual correctly see and perceive color changes as they go along. And again, I'm just going to display that these color change here is actually a smooth range. So the severity can be very precise, and clicking on the arrow will actually shift it about 2 nanometers each step. And you can also use the slider to get a little bit more precise with how much of the color efficiency you actually want to show. Alright, thank you for viewing. That's all.